goal of this video will be to discuss the cerebellum and specifically cerebellar ataxia. This is your brain, and this is you walking. Normally all the structures in your brain work together in a complex tango to initiate movement. I want to start by outlining how the brain works to control our movement in neurologically healthy individuals. Thanks to this structure right here, the brainstem, descending motor pathways are able to carry signals throughout the body via the corticospinal tract. As the signal descends down the corticospinal tract, fibers from this tract synapse with the motor neurons in the ventral or distal horns of the spinal root. This gives access to connections with the motor neuron pool where signals can reach motor end plates on skeletal muscle. At this neuromuscular junction, the neurotransmitter acetylcholine binds to receptors and triggers sodium and potassium ion diffusion across the muscle fiber, where subsequent depolarization triggers an action potential, just like in neurons in the brain. These action potentials initiate calcium ion release, which, without getting too much into it, bind to proteins on muscle filaments which aid in the actual contraction of muscle fibers and ultimately produce movement. That's all just to provide a brief understanding of normal brain movement interactions on a whole brain level. However, the brain is extremely complex, and even within the scope of simple movement, there is great intricacy. The main contributor to the coordination of movement is the cerebellum, which sits right at the base of the brain. Its gross anatomy is shown here, and consists of two lateral hemispheres, an anterior and posterior lobe, and vermis running through the mid-sagittal plane. Functional divisions of the cerebellum include the vestibulocerebellum, which involves vestibular reflexes and posture, the spinocerebellum, which projects to multiple spinal tracts and matches sensory inputs with output motor commands, and the cerebrocerebellum, which broadly coordinates movement planning. As we know, the cerebellum is essential for movement coordination and balance, and combined proper functioning of all its divisions is specifically critical for the regulation of these following processes. 1. Maintaining balance and posture. The cerebellum receives input from vestibular receptors and proprioceptors and modifies motor neurons' output in relation to changes in our body's position or muscular load. 2. Voluntary movement coordination. The cerebellum organizes force output and force timing across muscle groups to compose movement. And 3. Motor learning. The cerebellum helps with the fine-tuning and adaptation of our motor control to improve movement accuracy. While the cerebellum is largely responsible for executing control over our movement coordination, it receives help from the second-in-command vestibular system in order to do so. Much of the vestibular system is found within the inner ear, and its main job is to sense orientation and acceleration of the head in space. Resultant to changes of such, nerve cells transmit signals to the vestibular ganglion. The signals then continue through the vestibular nerves and are given to vestibular nuclei found here, who then transmit them to the cerebellum that is then able to process and make the appropriate compensatory adjustments to efferent nerve outputs about our movement. Everything prior to now was to provide a background in the regular functioning of our movement. However, this is where I will begin to get into cerebellar ataxia. Ataxia is a broad family of conditions that involve damage to areas of the nervous system that control and coordinate movement. Within the context of the cerebellum, ataxia is the atrophy of cerebellar cells and directly affects the inability of the cerebellum to contribute to normal movement functioning. This often significantly affects gait and balance, as well as motor mismatching synchronization. Let's dive a little deeper here. Ataxia can manifest in many forms, so just as to provide clarity, cerebellar ataxia will be the focus moving forward. Other types of clinical ataxia do exist, although challenges with movement coordination and balance are the hallmark signs of all of them. Etiology of cerebellar ataxia is not singular and is often split into acquired or inherited subgroups. Acquired ataxia could be caused by anything from brain tumors and injury to viral infection, whereas inherited ataxia is caused by gene mutation, most often presenting clinically as a form of spinocerebellar ataxia, where some kind of abnormal nucleotide repeat expansion is to blame. The literature provides lots of information as to the clinical presentation of this condition. The chapter in the Handbook of Clinical Neurology by Marsden gives clear indication of these symptoms and presents data on the effects. In this condition, the pattern of walking is notably abnormal. Strides shorten and the stance widens to increase support. Most often, gait variability will be decreased when the preferred walking speed is chosen and will be increased with greater disease severity. The gait is also distinctive in that it resembles more of a controlled falling motion, as the center of mass constantly moves away from the stance leg and is corrected by catching of the body by the swing leg. The Radmart et al. Paper seconds these ideas and adds that the cerebellar ataxic gait often resembles that as though the patient is intoxicated, as patients may struggle to maintain coherent stride paths and may even have challenges with remaining upright, often also experiencing clumsiness and jumpy eye movements. These angle-angle diagrams highlight a potential hypothesis, that being cerebellar dysfunction causes poor intralimb coordination, thus greatly increasing gait abnormality and variability. The left side shows the control patient, for whom we can see the overlapping gait cycles are relatively consistent over the different joints. However, on the right, with the cerebellar dysfunction patient, the gait cycles are quite different, and the patterns are far more variable. The implications of this are clearly seen in the gait, which we will look at in just a moment. Lesioning of different and specific areas changes manifestation of symptoms and is summarized as such. Vermal lesions cause ataxia relating to balance and gait, and saccadic meaning rapid and abrupt movements of the eyes. Lesions of the hemispheres cause ataxia relating to the ipsilateral movement of the limbs, and lesions of the flocculonodular lobe result in ataxia of balance and gait, and reduce the vestibulo-ocular reflex in terms of its suppression, gain, and adaptation. In the damaged state, the cerebellum cannot properly ensure we maintain balance and regular coordinated voluntary movements. The vestibular system, whose role we have established earlier in the video, normally projects signals from receptors that inform the cerebellum on the position and movement of the body. While vestibular ataxia is a separate condition, cerebellar ataxia affects the ability for vestibular signals to be processed. The internal computational model of the cerebellum, as shown here, cannot execute the compensatory motor adjustments required after encountering a mismatch in the expected sensory input and the experienced sensory input. Take, for example, the motion of hurdling. The anticipated sensory output would be the motion of your legs preparing to and clearing the hurdle, but if the experienced sensory input ends up being that you fall, the cerebellum cannot handle this mismatch. The cerebellum of ataxic individuals cannot anticipate the need for making these adjustments in regular human walking either, hence why their gait may appear clunky or sporadic. Signals that project the vestibular neurons are unable to be passed on, ultimately affecting not only the ability for the cerebellum to correct motor reactions in response to the unanticipated movement, but also the ability for the brain to take advantage of the motor learning properties of the cerebellum, and thus effectively update the internal computational model to normalize for the ataxic movements over time. Cerebellar ataxia is generally thought to be an untreatable condition, although fascinatingly, some research suggests that certain types may be receptive to some level of treatment. Much of this research discusses conditions with genetic origins for which ataxia is a present manifestation. For example, a person with ataxia resultant from deficiency of coenzyme Q10, which is caused by autosomal recessive gene coding mutations, may see some clinical improvement after supplementation with coenzyme Q10. Or ataxia with vitamin E deficiency, also a condition for which genetic mutation is the root cause, may improve with high vitamin E supplementation as well. Research around cerebellar ataxia has largely left out investigation into the motor learning aspect of cerebellar dysfunction. While research has been done to prove things such as the links of ataxia to Purkinje cell deterioration and cerebellar Purkinje cell activity to motor learning, not enough is understood about how these relations broadly affect our ability to predict and compensate for movement signal mismatching. Gaining knowledge about our motor learning and prediction abilities through various measured stages of Purkinje cell decay in ataxic patients would justifiably provide information that could be vital for understanding disease prognosis and progression in the context of compensatory movement strategies or for developing more appropriate therapies. Other research regarding the influence of comorbid neurodegenerative diseases would be justified. It's well known that other neurological conditions cause degeneration of cerebellar neurons. Think stroke, spongiform encephalopathy, and multiple sclerosis. Testing movement coordination, predictive movement adjustment, and general walking patterns in patients with the former conditions and patients with cerebellar ataxia may be helpful in determining similar or dissimilar symptom presentations. Such information could be used in fueling further research into the connections between diseases and could provide greater knowledge of disease markers. To summarize, the cerebellum is highly involved in our movement coordination and balance. It receives sensory input from many systems, including the vestibular system, which helps it to plan accordingly for expected versus experienced sensory input. In the disease state, as in cerebellar ataxia, the cerebellum cannot execute these functions, thus affecting gait, causing it to be more clunky and unorganized, and affecting our ability to form appropriate compensatory movement adjustments.